And kind of a funny story, I totally forgot that I'm rocking the mustache. So if you're used to seeing me with a baby face, I was surprised too when I hopped on and was like, had the camera on, is like, oh yeah, mustache on the face. And you might have caught in the email, if you saw those, that this is multi-omics layering. So what does that mean? Omics are systems biology. So like the microbiome, the metabolism, the genetics that we have in our body. And so nobody in the world has yet layered those successfully to be able to translate that data and put it in front of somebody in a really helpful, precise way. And so Dr. Jasbi has developed a test that is able and functionally capable of doing just that. So pretty incredible what he has done, and it's all done at home, which is wild. This really is like the next evolution of healthcare testing. I think this has the potential to become standard of care in testing for people because of what it can do, such as identifying disease signatures far before anybody else can, such as getting deeper insights for better, more personalized interventions, more personalized diet to people. And as you guys know, like it's really good just to have a general knowledge of health and how health works, but how much more valuable is it to know what works for your body personally? Because everybody's different, right? So if we can have data and analytics that inform us what is going to be best for our body, you know, some people don't respond to red meat very well, while others respond extremely well. And that's just an example, but having that level of insight to know what is going to be best for you personally, whether you're going through some chronic disease or you have cancer, is going to improve outcomes, improve your health, improve your wellness. And so I'm super excited to have this discussion with Dr. Jasbi today.
So we'd like to introduce you guys to Theriome, our very exciting and new biotech startup that we believe is going to revolutionize patient care, harnessing the power of metabolic testing and deep phenotyping with Theriome. So very quickly, this company was founded, really, the inception goes back to Alex and I's time in our doctoral program, where we really saw the challenges of low translational throughput between bench and bedside. And we decided to start Theriome to solve those challenges. Me, myself, I graduated from the College of Health Solutions with my PhD in 2022. Alex also graduated later that year after we had formed the company. I focus on mass spectrometry-based metabolomics. Alex is a leader in probiotic, microbiota, and the impact of dietary interventions. He is also a registered dietitian, in addition to his PhD. And our chief science advisor is Dr. Klein Theriome. Professor Theriome is a world-renowned expert in protein-protein interactions, systems biology, bioinformatics, and has been such an incredible guide for us on this journey. About us, as Mike has said, we are housed within Arizona State University. We're uniquely positioned between the intersections of academia and industrial testing. What we offer, and it was founded by Alex and I, to offer the first truly deep molecular phenotyping. The name Theriome, as it suggests, we integrate the theory of omics sciences, everything from the genome down to the metabolome. We believe in harnessing everything from state-of-the-art testing platforms to advanced artificial intelligence for analysis in service of that mission. And ultimately, our goal is to provide actionable results from the laboratory to our end users to improve their wellness. Our mission is very simple, and at the heart of it, we are a multi-omic testing platform. We leverage a wealth of curated scientific data across all levels of systems biology that really gives unrivaled health insights regarding customers' diet, lifestyle, and disease, as well as tailored approaches from field experts such as ourselves and you to achieve customers' health goals. We believe that every person is unique, and we're focused on helping you live better through the power of science. Very quickly, what we'll go through today, we're going to understand what our metabolic test provides. What are these deep insights? We'll talk about patient outcomes and potential financial benefits. We'll talk about our process, specifically our train by hands-on growth process. And then we'll talk a little bit about the current state of metabolic testing and where we stand as an entity in the field. We'll discuss the specifics of the Aristotle metabolic test by Theriome, and then we'll talk a little bit about the scientific evidence and that has really proven the concepts of metabolomic testing and single sample profiling. And then we'll talk a little bit about integration in clinics and discussing next steps. So very quickly, we at Theriome have the ability to unlock superior patient care. And of course, it's a financial growth with metabolic testing. They go hand in hand. The regular, complete blood panels are going to render anywhere from a couple dozen to a few dozen parameters on the blood. With just 2 dry blood spot samples, we're able to uncover the identities and concentrations of 126 aqueous sentinel metabolites that are central to human physiology and metabolism. And because we're able to multiplex those parameters via advanced artificial intelligence, as well as some nascent machine learning ensembles that we've developed, we're able to uncover really hidden factors that relate to very important health domains such as aging, mitochondrial stress, integumentary health, reproductive health, as well as things like environmental toxin exposure and mutational load. Ultimately, our goal is to boost revenue, improve patient satisfaction, increase referrals, and provide tailored treatments to our customers. Sorry, I'm just checking on the chats. Okay. Finally, I want to mention that, our partner clinics average $30,000 in health savings in preventative costs through our testing, and also average an extra $29,000 of annual revenue. So there's many ways where the Theriome test, the Aristotle by Theriome, can both improve your patient outcomes and increase practice revenues. So this is our simple process. First, we train, and that's pretty much you being here. And ultimately, our goal through this webinar is to give you enough information to confidently make a purchase. The next, of course, is buy. We will, in all fairness and transparency, we will attempt to sell you by the end of this webinar. And we only want you to purchase if you feel like it's a genuine tool that can be used for your patients and for the benefit of your clinics. Ultimately, the next step is hands-on. We have step-by-step instructions every step of the way, both on our testing kits as well as video instructions on YouTube and generalized instructions also on our website. And also we provide one-on-one guidance. When you purchase your test, you have a liaison with one of our members of our science team throughout the testing process. And so ultimately, our goal is for you to incorporate this Aristotle test into your practice, simultaneously enhancing patient satisfaction and outcomes, as well as revenue. So let's talk a little bit about traditional blood tests. So these have been the cornerstone of metabolic testing for decades. However, lots of the associations that they've been proven are generalized associations, and the parameters are not biologically relevant enough or numerous enough to be able to tailor approaches. It is, far better to understand what kind of person has a disease than what kind of disease a person has. And so these traditional blood tests really offer a very narrow view into a patient's overall health status. Like I said, generally returning a few dozen parameters. Genetic tests, which are, of course, more recent developments in health testing, provide insights into an individualized predisposition. But most conditions are not genetic conditions. They're rather genomic conditions, and their etiologies are very heterogeneous, meaning that the gene is not solely to account for it. So genetic tests ultimately, for most conditions, especially the ones that contribute greatest to human mortality and morbidity, those conditions is incredibly heterogeneous. The genetic tests only assert very broad probabilities of ever developing a condition, whereas a metabolomic test provides a current snapshot of that very dynamic system. And that brings us to metabolomic testing. Metabolomics, which was started in the late 90s, but traces its roots back to the early 70s, really, is a science, an interdisciplinary science that is the intersection of analytical chemistry, physics, biology, and bioinformatics. And our goal is to study and characterize the metabolites in the body, of which there are tens of thousands. And we have selected in our panel 126 sentinel metabolites that provides excellent sensitive coverage for the vast majority of human pathways. Theriome is reshaping what we consider to be healthcare. We use our proprietary artificial intelligence to analyze patient-reported demographic data and biological data that we collect via our mass spectrometry and sequencing platforms. We create a unique snapshot of that patient's health, and that AI offers personalized recommendations based on the backgrounds that these patients have reported to us. We're also preparing to integrate blockchain for transparent and interoperable data handling with existing healthcare systems. And so this harmonious blend of leading edge technology and rigorous science really places us at the forefront. And there is, by all measures, no metabolic test on the market more advanced than the Aristotle. So before we get into metabolomics and the Aristotle test, let's just have a brief refresher of systems biology. So the genome is the entire set of the DNA that is found in all cells. The transcriptome would be the full set of coding and non-coding RNA, essentially what's transcribed from the genome. And then the proteome, one level after that, are the pieces that are translated to proteins. What RNA is expressed to form protein complements of cells, organs, and organisms. And all the isoforms of those proteins are collectively referred to as the proteome. We then go to the community of microbiota, and the microbiota is not just, bacteria, it's archaea, it's fungi, it's viruses, and that microbiome really is itself its own pyramid, interacting with your own systems biology. And then nearest to the phenotype, down on that hierarchy, is the metabolome, which is the global set of all molecular weight products of all upstream processes. We liken this to, baking a cake, where the genome is the set of instructions, the transcriptome is the availability and pricing of the cake ingredients at the supermarket, the proteome is everything you brought back with yourself from the supermarket, minus what you may have dropped along the way on your walk home. The microbiome could be considered, the quality and precision of your cooking instruments, your baking instruments, and then the metabolome, just like that cake, is the complex product of all of its upstream processes. And as you notice, it sits at the nexus between the environment, which is immediately below it, and the genome, which is above. So for most diseases, of course, that are heterogeneous in their etiology and are due to a combination of genetic and environmental causes, which is most diseases, right? Heart diseases, cancers, neurodegenerative disorders, et cetera. These are a combination, and so we want to target that intersection of the gene-environment interaction, which is the metabolome. So the potential applications of metabolomics over the last 30 years have been myriad, and on the figure to the right, you can peruse some of the basic and far-reaching applications that metabolomics has enjoyed. We've named our test after Aristotle, and of course there's this famous quote from the Nicomachean Ethics, knowing yourself is the beginning of all wisdom. And we named it Aristotle, not just because of that quote, but he was the first person really to theorize on the concept of metabolism and commit his thinking to writing. Essentially, Aristotle knew that there was something in the food that when we ate replenished the basic building structure of the body in addition to generating heat. So he even had a nascent understanding of entropy. And we named our test after him as an homage. And so metabolomics, like I've mentioned, is the study of all small molecule products, specifically anything that's less than 1,500 Daltons. And we, as I mentioned, it provides a direct functional readout of current physiology rather than, broad probabilities of what could be happening at the genetic level. And so we balance coverage and sensitivity, and we provide our customers with this very dynamic snapshot to better inform their health decisions. Like I said, it's nearest to the phenotype and provides the most detailed biological readout. The metabolome is therefore a very attractive target for testing. We couple it with mass spectrometry and chromatography to get high sensitivity, wide detection range, as well as a wealth of database equity that will essentially substantiate and provide fidelity to our results. Metabolomics, if you've not heard of it, no worries. It's very up and coming. If you go and do a PubMed search of metabolomics and studies over the last 20 years, you'll get 37,000 results. But like I said, it's very fast growing. We've recently hit an inflection point where there's critical interest has been achieved in this field, and clinicians and researchers from other disciplines are now incorporating metabolomics into their practice and their science. So of those 37,000 studies, 24,000 were published in the last 5 years. So much of that interest has been recent. It has facilitated numerous discoveries related to biological diseases, even behavioral disorders, and is commonly used in the drug development space for pharmacokinetic and pharmacodynamic studies, as well as monitoring of clinical interventions and safety and efficacy trials. And in conjunction with multivariate statistics and advanced machine learning algorithms, Theriome is in a position to provide a much deeper understanding than just what the data by themselves represent. The data by themselves, without critical advanced bioinformatics, are just numbers in a spreadsheet. And so in conjunction with our bioinformatics pipeline, we do give patients a much deeper report than what is currently available. A very brief overview of how this test works before we dive into some more specifics. You have a finger prick test that is put on a dry blood spot. It is shipped to our sample, to our central lab for processing. We use gas chromatography mass spectrometry to sort our metabolites by retention time and mass to charge. And finally, we multiplex those parameters, those 126 central metabolites that we captured in our assay, and feed them to our artificial intelligence pipeline, which spits out a user report. So how does it work? You are the source of the metabolites. Your gene environment interactions are the source of the metabolites. We go through a process once you receive your sample of quenching, grinding, extraction, coupled with derivatization. We'll see some photos of this in the coming slides. We first separate, we do a first dimension separation based on the chemical structure using gas chromatography, whereby molecules will interact variably with the stationary and mobile phases of that chromatography column. And then after sorting by retention time, we then sort in the mass analyzer by mass to charge ratio. And how this works is that your sample comes in here, riding on a stream of helium gas, of inert gas. The sample is vaporized. It's hit with an electron beam to essentially volatilize and ionize your metabolites in the dry blood spot sample. It's accelerated through the ion optics, and then using a combination of radio frequency and AC/DC charges, we are able to send your metabolites to the detector. We take your chromatogram and a spectra and quantify the area under that curve, which gives us a concentration for that metabolite in that customer's blood. How does it work? You receive a dry blood spot kit in the mail. You collect it in the comfort of your own home. Once complete, you ship your blood card back in the biohazardous envelope with a desiccant pouch. When we get that envelope back, we start processing it. First, we undergo homogenization of the sample as well as protein precipitation. We then sonicate your samples at minus 20 degrees Celsius to essentially further separate any metabolites from that paper matrix. We then undergo ultracentrifugation at 22,000 G, 4 degrees Celsius for 10 minutes to separate layers. We then take that separate aqueous layer, as you see in step 9, and we extract that to a new tube. Then we take that aqueous layer and we dry it to form these residues. After we formed metabolite residues, we reconstitute them to form and derivatize them on the heating block to form various trimethylsiloxane derivatives. This essentially volatilizes the compound for flight in the chromatography tube. Once more, we centrifuge our samples to ensure adequate cleaning and reduce any potential for decreased ionization efficiency in the electron ionization source. Then we take the top layer and we extract that for GCMS analysis. So very quickly, samples are placed in the auto-liquid sampler arm, and we load our proprietary gas chromatography mass spectrometry method. Once the sample has been injected, each sample takes about 30 minutes to run, given the various chromatography ramps that are happening in that machine's oven. Essentially, we ramp the temperature from 60 degrees Celsius all the way to 325 to variably elute our samples from on the column. And then we integrate, we identify, after mass spectral filtering and sorting, we identify the identity of those metabolites by their retention time and mass to charge, all of which has been confirmed using a pure analytical standard of that compound. Peaks are algorithmically checked. They're automatically filtered for quality, and then they are exported to CSV for analysis. Our current bioinformatic pipeline is performed in R. A total of 66 lines of code are implemented to produce these automatic bioinformatic results that are mapped to the individual's profile, and the report is generated. Here's a description. Here's the actual machine we use. This is actually in our lab there. And what we do is we provide excellent sensitivity with metabolome coverage. There's many platforms that can be used to detect metabolites. You can do kit assays at the bench. You can use nuclear magnetic resonance. NMR is another major platform in metabolomics. Unfortunately, it doesn't provide the sensitivity that we would need to adequately detect the very low concentrations of metabolites that are present in just a few drops of blood on a dry blood spot sample. So we don't use NMR, thankfully. We use this alternate platform, known as mass spectrometry, which calculates mass to charge ratios, and it provides 10 to the 6th times more sensitivity than NMR. And with it, we're allowed to achieve 126 metabolites separated on column. Here's our platform if you'd like to learn more about agile instrumentation. And it's suitable for detection of various metabolites in these panels: exogenous, endogenous, food, plant, microbial metabolites, drug metabolites. And we've also made sure to include in our panel metabolites relevant to toxins and pollutants as well as cosmetics. And so we have various sample prep methods. This sample prep can be augmented in certain ways to allow for different biospecimens. And currently we've optimized it for the detection of dry blood spot samples. So at the end, we produce 126 metabolites. And like I said, I keep saying this word sentinel metabolites because these metabolites recur in various pathways. And for us to say that we have reliably detected a pathway, we need to be sure that we're measuring at least 2 byproducts in that pathway to form a real set. A single metabolite does not constitute a pathway. And so these metabolites are redundant, and we essentially take advantage of that biological redundancy that's inherent in human metabolic functioning. And we've selected these 126, which provide a great overview of general human metabolism. It probes 14 different chemical classes. As you can see, most of our metabolites in the panel are amino acids, as well as saturated fatty acids, dicarboxylic acids, and TCA acids related to TCA acids, pyrimidines, various amino fatty acids, and some branched fatty acids and unsaturated fatty acids. Some very important subclasses that are biologically germane. And as I've mentioned, when we map our results, and this is a representation analysis of our 126 metabolites, and we've performed a network view of them. And as you can see, the pathways of the 99 pathways in human metabolism, 84 of them are reliably detected. We can monitor 68% of all human metabolic pathways reliably using our Aristotle test. It's very orders of magnitude greater than the metabolic coverage that you get with current tests. And we produce, and here are some sample reports, and this information is also available on our website if you'd like to take a closer look. We produce, our Bayesian AI generates health scores based on your 126 sentinel metabolites, as well as the demographic information we collect to produce your optimal scores across 12 key health domains from 1 to 10, 1 being not optimal, to 5 being neutral and 10 being optimal. And those scores are aging index, nutritional index, inflammatory score, mitochondrial health, cardiovascular health, reproductive health, liver and gut health, neurocognitive index, and tegumentary health. And as I've mentioned, an environmental toxin exposure and mutational load, which a lot of our customers have felt really useful. In addition to the scores that we provide, every score comes with personalized recommendations based on what metabolites were altered in that set, what the degree of change was, what the direction of change was, and how that relates to the demographic information that we've collected from you prior to testing. We also query your results against 344 documented blood signatures of disease. And so because we have a database of 344 disease signatures and the metabolites and the relative changes from control, we're able to give you, for instance, this as a sample report. We have 26 metabolites that are related to schizophrenia in our panel. Of those 26, this customer had 6 of them that matched in direction and magnitude of change. And we provide a match percentage for this. We provide a match percentage here for that metabolite set, which is 23.8%, and a confidence in that match percentage. The confidence being the analytical error that may have been introduced, and the match percentage being the objective overlap between this customer's profile and the known metabolic profile for schizophrenia. And like I said, we have 344 disease signatures, and we flag anything we see with a confidence greater than 95%. So this patient has some things related to early markers of myocardial injury or heart failure. How does this essentially work, and how do we return metabolic set? How do we identify biomarkers? How do we build this database? And how do we turn that toward customers and make this a patient-facing enterprise? So I published this back in 2021, and I was giving this talk at the Arizona Physiological Society in 2019. And what we did was we took samples from the BSHRI, which is the Brain Research and Health Initiative from Banner Health, which is our local hospital here. We took the neocortical tissue, specifically the frontal gyrus, of patients who had various levels of this disease. We had 48 patients, 12 that were normal controls with no evidence of pathology or symptomology. We had 12 patients that were high pathology controls, meaning that they had the neurological, amyloid beta and neurofibrillary tangles that are indicative of Alzheimer's, but they had no symptomology. Then we had mild cognitive impairment individuals whose brains were obviously collected postmortem, and those people had the pathology, the symptomology, but did not progress to full diagnosed Alzheimer's. And then finally, we have samples from 12 frontal gyrus samples of patients with confirmed postmortem Alzheimer's disease. And as I mentioned, we go through the extraction protocols, different, but very much similar to what you've seen in the process of homogenization, quenching, extraction, derivatization. In this study, we used a combination of gas chromatography and liquid chromatography, and we essentially go through the same biomarker pipeline we would at a research study, and we apply this to our customers. And we were able to find some very interesting biomarkers and effects of the metabolism in Alzheimer's, which again, we published this in 2021 in the Journal of Proteome Research. Essentially, we found 4 very diagnostic fatty acids: lauric acid, myristic acid, stearic acid, and palmitic acid. And as we can see, they had variable relationships between the groups. As pathogenesis increases, we see an almost linear decrease in lauric acid, whereas the relationships with the other 3 are a little bit more stochastic, going up and then down. But we still see that we have 2 relatively typified models here in relation to disease progression. When we performed a random forest analysis with 500-digit decision trees and randomness turned on, and we took that analysis and we looked to segregate our patients, we found that we had a 91.7% diagnostic accuracy between our Alzheimer's high pathology control, mild cognitive impairment, and normal control groups. We also took that data and we queried it using our enrichment database. So these are tools that are available to researchers. We basically take the findings of other studies, known metabolite sets such as lysine degradation, fatty acid metabolism, and we look at impact scores and significances. And as we've seen here, we were able to see that 3 pathways in between these people with and without symptomology. 3 pathways were highly impacted, specifically the linoleic acid metabolism and the metabolism of those amino acids, whereas 3 pathways had high significance and relatively low impact: glycine degradation, fatty acid metabolism, and the branched chain amino acid degradation. And what we did was we combined that with our enrichment analysis. Here it's visualized as a network view where we conducted it between our cases and our controls, and we found 11 different enzymes that were significantly predicted to be different between those groups. Since we're measuring, in this case, we're measuring thousands of metabolites, but even with 126, we're able to infer predicted changes at levels such as the enzymes, at levels such as SNP loci. And we integrated our findings really to create this synthesized hypothesis of what we believe is happening. And what we saw were progressive changes in substrate utilization and energy production as patients progressed from normal controls to high pathology controls with mild cognitive impairment. We saw a shift in substrate utilization from glucose via the GLUT1 pathway to the branched chain amino acids in all of our high pathology controls, and roughly half of our mild cognitive impairment group exhibited this shift down to energy utilization via branched chain amino acid degradation. And then roughly half of our mild cognitive impairment individuals, as well as all of our Alzheimer's disease individuals, appear to have a preference for fatty acid utilization, bringing it in via CAC-T and finally beta oxidation for the generation of ATP in the electron transport chain. So this is just a nascent view of how we take the neocortical tissue of people with known conditions and classifications. We analyze that metabolite data using a combination of our analytical chemistry and bioinformatic techniques, and then we interpret that to the existing pathologies we know and essentially tie this bridge between the molecular level and the sort of pathogenic level of amyloid beta plaques developing. And you can read the study as well and see how exactly we made that connection. But a lot of those metabolites that we identified were indeed related to amyloid beta plaque development. Micah talked a little bit about multi-omics layering. And that's exactly what we're doing here. My co-founder, Alex, is developing the BioMe test, which is currently not available, but it is the most advanced long-read 16S stool test, which is conveniently collected from a bunny wipe after you have had your expression. Instead of usually a fecal tool collection kit, you never have to handle anything, and it's very convenient. It's the most advanced long-read 16S test that will be on the market soon. And this is a concept that we kind of developed in our graduate programs. This is a study that we published just this year or last year, Last year. We published this last year. As we were graduating, we took a class together. He actually emailed me and said, this class looks great. It was called like microbiome data science. And it was taught by this incredibly advanced and well-respected professor. And we both decided to take it. And in that class, he really stressed that we should bring data to the course whereby we could learn how to analyze, but also potentially publish on. And what we had were data related to screen time exposure of college students here at ASU. And we had both their 16S amplicon sequencing as well as their mass spectrometry data. And we were able to layer this, not just through basic correlation, but through machine learning co-probability analysis to come up with a very cogent hypothesis of what was driving the observed effects between those with very high screen time, more than 75 minutes a day, and low screen time, less than 75 minutes a day. And what we found is, as you can see, these are the taxa that we found between the high and low groups, and general differences between these various species. We also here looked at the PCA generated from the dicode analysis. This is Alex's work on the manuscript. And then for every subject here, we have these bar graphs that clearly show the relative abundance of these taxa for each subject in the high and screen time group. We also found metabolites that were related to high and low screen time. These were all significantly different between our study groups. And on the top, you see the box plots of these 5 significant metabolites. And at the bottom, you see a heat map whereby you can clearly see generally higher expression of these metabolites in the low group and generally low expression for half this group with regard to proline and alanine, and lower expression for 1-methylhistidine and the others, generally speaking. And these produce significant results between the groups. So we layered our results using this technique known as MMVec. And maybe Alex, do you want to speak a little bit about MMVec?
So one of the difficult parts of bringing together these different data sets is not just doing normal, what in science we call correlation, or basically association. It's understanding when and how these are co-occurring. So we have an advanced machine learning process that we utilize where we can actually detect these across various different samples and various different metadata. So if we're concerned or we're looking at a particular disease state, or in this case, screen time, we can detect differences in co-occurrences of these specific metabolites, and then also specific taxa, or basically like what we're doing is gut bugs, right? The species that are inhabiting your gastrointestinal tract. And from that, and Paniz can speak to this, we've formed this kind of conceptual framework for how they're kind of operating together and what type of biological effect they might ultimately have, because that's what we're really concerned about. When you profile the gut microbiome, you're talking about the gut microbiome, but what we really want to know is how that is affecting our own physiology and our own health.
Exactly. And so when we layered our analyses from our metabolomics and our microbiomics experiments, as you can see, these are more than just correlational estimates. We know that in the high screen time group, these metabolites, these species on the right here, these were expressed in greater relative abundance in the high screen time group. And we know that they all consume phenylalanine. And so phenylalanine, we know, is reduced in the high screen time group. Phenylalanine is a gut metabolite. So we know it's not in the intestines, not here in the enterocyte. And this is just the inset, as you can see. This is the inset of a enterocyte, what's really happening in that mitochondria of that enterocyte. So phenylalanine is in the lumen, having this variable consumption and production with these species. And we know that this phenylalanine is coming in lower than it should. So tyrosine, its metabolic downstream product, is also lower. And as it's being plugged in, these are the metabolites that we have seen in that metabolomic screen. And if it's green, it's gone down in the high screen time. And if it's red, like GABA, it's gone up in the high screen time. Additionally, we've layered the inferred changes that we saw at the enzyme level, at the protein level, specifically glutamate dehydrogenase and this S3 amino 2 methylpropionate transaminase enzyme. And although we didn't measure these enzymes, because we have so many measurements around them, we can infer that there's a significant difference in this dehydrogenase enzyme. And our data is showing that there is a very high significant difference in this enzyme. And of course, these enzymes are embedded in the pathways that were already indicated by our other analysis. So it's really, this is the power of AI, is that it figures out in a very layman terms where all the fingers are pointing. And then finally, and I'll go through this very briefly, this is the most recent study I published just last month. This is in Neuroscience, a Springer journal. It's not open access. So if you do want this one, just let us know and I'll be very happy to share the PDF with you all. We essentially looked at the metabolomics of aging. Aging is a central process to disease. The number one risk factor for disease is age. So understanding what is normal and healthy aging is of paramount interest in the molecular biology community. So we undertook the same study design. We looked at people who were young and old, 75 in the young group, 76 in the old. We analyzed their serum, this time using LC tandem mass spectrometry. So this is chromatography that is carried out on a liquid reagent, and we produce 2 MS spectra. We do the same analyses to render, again, biomarkers that were found to be significantly different between the young group, which is 21 to 40 years old, and the older group, which is 65 and older. And these are coming from our general linear model that has been already adjusted for sex, BMI, and chronic condition score, essentially all of the confounding covariates that might mediate or moderate the relationship between age and these metabolites. And again, I skipped a couple of steps for this last case study, but we were able to layer our univariate results, our multivariate results, as well as our predictive machine learning algorithms to come up with this very synthesized understanding of what's going on. And compared to the older participants, the young group displayed greater abundance of metabolites related to lipid and nucleotide biosynthesis. And in the, so the age restrictions I saw that comment. The age restrictions are just 21 to 40 years old and 65 years old or older. That's what we defined as young and old. And yes, there's a gap, but there's a scientific reason for that gap. It's basically to assess the extremes of age first before we do a more continuous analysis to see if those extremes are maintained in the middle.
I think the age restrictions was a question towards, is there an age
for
you have to be to take the test?
Right. It can be 18 years old, or if ordered by a parent, it's also a parent is able to consent for their child. Parents or legal guardians can order the test for their children. Yes. And so again, we were able to layer these results. And so somebody asked earlier, and I just want to iterate before we leave the science, and I'll just leave this here for people to look at, how is this different from Viome? That's a great question. Viome, do you want to talk a little bit about what the Viome is?
You go ahead.
Okay, so Viome is essentially a gut microbial test. It does a short read 16S, which is a very non-specific form of testing, to look at the bacteria that is in your gut. And I know they do a saliva test as well, and maybe a skin swab. But that bacteria is not your metabolic functioning. That bacteria is not the level of the metabolites. We study very, very small molecules. Like I mentioned, less than 1,500 Daltons. That's like 1/1000th the size of let's say, a protein.
I want to go back to that systems biology pyramid to explain where the metabolites or the metabolome fits in this whole.
Yeah, so if you remember the systems biology pyramid we had the genome, the transcriptome, the proteome, and the microbiomic, which was in that layer, but as I mentioned, should be its own pyramid, truthfully. That's just a collection of the bacteria that lives in you and on you. These metabolites are what interacts with that. So if we go to this slide, these are the bacteria, and this is essentially what Viome is measuring, and what we will measure in a few months with a much more advanced and analytically rigorous test. The test they use has fallen out of fashion from academia for the last 5 years at least. what we are offering is the measurement of these metabolites. These metabolites are more able to speak to things like enzymatic functioning and pathway health. These bacteria really are interesting when you have the metabolomics data and you can layer and fine tune the results.
At best, you from different sequencing methodologies, you can infer function. What the metabolome offers is actually validating function. So right now, there aren't any tests that we're aware of that fully bridge together the microbiome and the metabolome to get a full picture of form and function. Because ultimately, that's going to really matter when we're talking about forming these different health states, or as Paniz showed with his what he's producing for your reports, different indices of health, which is, for the normal consumer, a lot of this science may not make a lot of sense, but if you're able to see this in a simplified format with actually having tangible takeaways, that's when this really becomes powerful.
Exactly. And one of our key health domains that we report on is the gut health index. And that gut health index is calculated based upon known beneficial metabolites that are produced by bacteria and known harmful metabolites that are produced by bacteria. And so by measuring the lower level, we are always able to measure to levels above. But the case is not the same if you are going down a level. So if you measure the gut microbiota, there's no guarantee that those biota are functioning. You can get a compositional awareness of what's going on, but the function remains ambiguous at best, whereas the metabolome is a direct readout of current physiological function.
And I have a good question here. It says, should I wait for the more sophisticated tests coming in a few months? But one of the cool things about this multi-layering is that you can do the Aristotle test and get this really robust analysis of what's going on in your body. But that data can be compounded with a microbiome test. So once the microbiome test is taken, you can available, you can take that test, automatically can be combined with the Aristotle test. So it's not something you have to wait for that to come out. It's something that you can do at a later date, and it'll still apply to the test that you've already done.
Exactly, and that's a great clarification, Micah. Thank you for making it. And I'm sorry if my language introduced some confusion. The more sophisticated test I'm referring to is the one that we're developing that is more sophisticated to what's currently offered by Viome and other gut health testing entities in the space. As we would like to think, vastly more sophisticated. What the test, however, that we're developing, the Viome test, should not be viewed as more sophisticated to the currently available Aristotle test. It's in fact complementary and will only enhance the insights we get. In fact, we get more insights by doing the combined analysis of these two layers rather than either one individually. And so, diagnostic testing, I want to highlight this because it's something I've personally been more interested in as we bridge the gap from research to patient-facing data. And I've reassessed the merits of diagnostic testing and what it means. And so looking at this study that was published by some researchers from the University of Rochester School of Medicine, patients often equate testing with thoroughness in medical care, and they tend to view doctors who order more tests as being more thorough and careful. And this other study published in the British Journal of General Practice found that 90% of people, it's staggering, want their doctors to offer all medical tests possible, and 85% want their decisions about their care to be made with their doctor. In this really great piece, I would encourage everyone to read. This is published by Dr. Berry in the New England Journal of Medicine. It states, it's an opinion piece, but it states very clearly that patients view diagnostic testing and collaborative decision-making as a very important aspect of their care and actually a reflection of their provider's competence as well. And in this Cochrane review of 86 trials that was published back in 2009, the patient's decision aids are the use of them are, leading to increased knowledge by the patient, more accurate risk perceptions by the patient, greater number of decisions that are consistent with that patient's value, reduced levels of internal conflicts, and fewer passive or undiagnosed patients. So this is our offer. We hope you would enjoy a special discount today. I'll let Micah handle this part a little bit more, but we have a special offer for our webinar attendees.
Yeah. So I don't know if you guys, Paniz is tough to keep up with, right? Really brilliant scientist guy, has a lot of fantastic information, but in a nutshell, what he has developed is far deeper in terms of insight into how your body is performing and what it's doing. And are there any diseases that haven't turned into symptoms yet that you might not know about? So it's giving you a really good picture into the state of your body and how it's working. And it includes things like your biological age and your liver function and some really good stuff to understand. So it's like seeing the future of this. I'm hugely excited because I think this has the potential to become standard of care in the future, meaning that, hospitals are utilizing it, that it's one of the first tests that is done when a doctor thinks that there's something up that they need to understand with their patient. And so I think this is going to be a massive revolution in healthcare, getting this test off the ground and seeing it go. So if you guys are interested in participating in all this 10 years of science and research that Dr. Jasbi and Dr. Alex have done, then I would highly recommend taking this test. I've done it, and I was actually a little bit relieved because Dr. Jasbi was like, well, your test is kind of boring. So I was like, well, that's a good sign. It ain't, I don't have anything going on, right? But yeah, nonetheless, it was really good for me to understand where my biological age is, where my liver function is at, and where, how my body is doing. And then over the years, I'm going to continue to take this test. It's going to get more sophisticated, but that's also going to be indicating to me, are the things that I'm doing putting me in the right direction, pushing me in the right direction of where I want to go, of being healthy and thriving. So if you guys want to participate in the science and what Dr. Jasbi has put together, we can actually ship the test kit to you. You collect the sample, which is a little finger prick. You put it onto the dry blood spot sample. And then there's a label in the box that you ship it back to Dr. Jasbi's lab in Phoenix. And then within a couple of weeks, you'll receive a report that tells you all this data, all this wealth of knowledge of how your body is actually performing. So for Dr. Jasbi normally has this test at $830, but we whittled him down to give us $499 on these tests. So he's actually offering $331 off. That's going to expire, I think 2 days from now. I can't remember exactly. So today is Wednesday. Yeah, Friday night it expires. And so I'd highly recommend jumping on this. He also only has 39. It's 39, right?
Yeah, we have 40.
40. Okay. My bad. I've been saying 39. So we have 40 he has 40 materials left to do samples until his next batch comes in because he's got to order some more supplies. And I'm not sure how long that takes. But at any rate, you guys, if you want to do this test, there's 2 ways that you can order. You can drop your email into the chat. And just put your email there. We'll send you a quote and instructions on how you can take this test and participate in this. The second way, again, discount expires in 2 days, and it's with the code TESTME, is you can go to Theriome.me. I'm going to put it into the chat here. And check out on the website there if you prefer to do it that way. We make it easy for you. If you drop your email, we'll send you the quote along with the instructions. A little bit easier, a little bit faster. So feel free to do that if that's what you want to do. Or you can go to the website, therio.me, to get one of these tests. Again, there were 600 people that signed up for this. There's only 40 tests available right now until he gets his some more materials. And, I'm really excited about this because it's very like grassroots. So Dr. Jasbi is out of academia, and he had this moment of realization. I'm not sure when it was, but I know we talked about this, that you're like, academia is not the channel to get this test out there. It has to have some monetary incentive behind it because it's just going to get lost in the annals of academia if we don't do something and turn this into a company. But that being said, I'm very excited because this is like the very beginning of something that is going to be massive, that's going to be revolutionary to healthcare. You're going to get much deeper, much better personalized medicine. Like I said, understanding what is working for you, or if you have some sort of chronic disease or some sort of issue going on is like, am I going in the right direction objectively? Not just do I feel better or do I feel worse? Objectively, is my body responding to these things in a very positive way? And then also for me, just for peace of mind, understanding like, hey, do I have any sort of cancer runs in my family? So like, that's something I think about. Do I have any underlying issues going on in my body currently? And he can actually develop what's called disease signatures, which are these metabolites that are associated with different disease states. And he can produce a report that says, yeah, you're at this stage or you're at this stage. And it's way earlier than anything else can provide. So again, I know this is long-winded. 2 ways to get this test. One is drop your email in the chat. Number 2 is go to therio.me. I'm going to drop it in the chat again. And you can use the code TESTME to check out there. It expires Friday evening, so make sure to do it quick. And yeah, that's the synopsis. I really appreciate everybody jumping on here, but it looks like we have a number of questions. So Dr. Jasbi, what do you think about going through some of these questions?
Yeah, absolutely. Yeah, I was just typing my response to someone who said, there's so many genetic tests on the market. What makes this different? I want to just stress that we are not testing the genome. Although we have the ability to infer 4,598 S&P loci at the genome level, we are not testing, base pairs. We are measuring metabolite levels, so the expressed downstream products of those genes. If you go to test, your BRCA1 gene for breast cancer, you might get a probability in the range of 20 to 80% that you'll develop breast cancer, which is no better, if not maybe worse than 50-50. Whereas a test like a metabolomics screen, such as the Aristotle, can provide that downstream dynamic. We are sampling the layer at which gene-environment interactions take place. Any research and healthcare professional will tell you that the genetic tests will take you only so far, and it's not really that far. The promise of the Human Genome Project was this revolution in care and patient care and research that we haven't seen yet.
Well, usually you're just taking that test one time.
Right.
So as your health state varies, the metabolomic test, the Aristotle, can more accurately capture that, because our hope is that we can change some of these states, right? It's not that we just profile the genome and we say we have a greater probability for this disease, et cetera. It's that we can actually take control of our health and do something about this.
Exactly. Nobody is their genome. If we were, we would all have a genetic test. We would have whole genome sequencing done this afternoon, and by tomorrow morning, every disease and ailment would be cured. The idea is that the greatest contributors to human morbidity and mortality exist at the nexus of gene-environment interactions. Their etiologies, their causes are so heterogeneous. They don't come rarely just from the environment or rarely from your genome. It's very organized and sophisticated interplay between the effects on the body, the stresses downstream, and the genomic influences upstream. And that's what we measure. The metabolites are a direct physiological readout of that interaction space.
Sorry, keep going.
No, go ahead. Well, I was going to go to the
really
questions.
Good question here. So Brent asked, it seems like this product will demonetize some powerful medical players. I've heard that inventions with that aim tend to be bought and then suppressed. What are your thoughts on open sourcing the tech? So maybe take that from a higher level and give some sort of response to that, because I think that's a great question.
Absolutely. Yeah, go ahead. I think you have a great.
Well, yeah, I think what we're doing from the very beginning is bringing in blockchain, bringing in AI. I think these are going to be protective mechanisms for our company, for the data that we're going to acquire, and to keep things open source and highly transparent, which I think will help safeguard against some of those issues.
Exactly. Our future plans to integrate, blockchain are going to give us extreme data transparency with who has accessed that data, what entities it's been shared with, a very detailed audit trail, immutable source of information, as well as autonomy put in the patient's hands. You will control your own data. We understand that, technologies that aim to do what we're doing do get bought and shelved. we're in no hurry to sell. And our goal ultimately is to create a testing entity with a body of knowledge that is capable of answering all relevant medical questions, at least in the sense of directing future research. Keep in mind that these are associative analyses. These have to be proved with causal mechanistic research, but at the very least, we are providing an atlas to human health and functioning and a roadmap to what researchers should target next, and specific enzymes, metabolites, and pathways with the relation to specific disorders as we have analyzed in our data. And just to kind of give you guys an idea, this is the beautiful building we're in right now. Actually, our physical lab space is on this third floor. So this little section here, this is our window lab space, and we are in a conference room just around here. And so come visit us in the heart of downtown Phoenix. This is our laboratory address, and this is our mailing address for when, if you order your tests, you want to send back your samples. Of course, this information is given to you in the testing kit. And now I think we can go to the questions. What do you think, Micah?
Yeah. I mean, we've been kind of answering some of them as we go, but just another comment on that transparency aspect, the open sourcing idea. Dr. Jasbi is thoroughly transparent. I mean, he had me in his lab one day, let me record as he explained like everything that he does and things like that. He does have like some obviously proprietary knowledge of how this test is translated and how this data is translated. But as we grow and as it gets bigger and bigger, there will be more people with an in-depth understanding. There'll be AI involved. And I think it's really exciting to see this, because I think we're, keeping the end in mind to a degree of like, hey, we don't want this test to get suppressed. We want it to be a benefit to humanity, not just another one of those things that kind of gets lost. And so it's definitely being built with that in mind.
And I would also like to stress, when we talk about open sourcing the platform, the blockchain will be an open source project. The AI is a proprietary developed algorithm that is of our creation. The testing platform, however, is already open sourced. Mass spectrometry has been around for 150 years. Of course, it's developed from J.J. Thomson's initial detector at Oxford University to what we're using today in modern mass spectrometry clinical labs. But we actually do have open source platforms. None of this is proprietary to us. Sequencing tech is used by every university, every science department in the country. Mass spectrometry is a mainstay of every research institute in the country. The difference is that these platforms are not at your hospital. These platforms are not at your Sonora Diagnostic health testing clinic. These are research platforms, and many times they occupy have a considerable footprint, and require very advanced, specializations from people with doctorates to operate them. So you won't find them in LCMS OAT. Mark.
Another good question here real quick before we get into some of the other ones is,
Sure.
How can physicians learn how to use the results to help patients? So how do we take this test as a practitioner, medical practitioner, or maybe you're a patient and it's your doctor that you want to utilize this test? How does that practitioner get an understanding of using this test to get better outcomes?
Right. So our results are given in three different ways. And it is intended to be digestible by our direct-to-consumer customers, as well as by the clinicians who order our lab. How do we do that essentially? Our results are given in very easy to explain language. Although it's scientific and we use some scientific terminology, we always take care that in our reports it's structured in a way, not just presented, but the content is structured in a way that it's easily digestible information. That's the first page. That's the index that I believe a customer can read and fully digest by themselves. Not I believe. We've had it, and the customers have said this is very interpretable. This is the first time I feel like I'm interpreting my health data in a meaningful way, and I'm actually able to approach it. The next pages of our analysis, of course, are the recommendations. And those recommendations are very much in line with, what a physician would recommend. It is trained on generalized recommendations for specific metabolite levels and altered given the demographic information that we collect self-reported from our customers. And then it's giving you very easy to digest recommendations for each domain, bulleted points that are easy to read and easy to implement. We are trying our best to demystify the science of metabolomics. Essentially, this is a huge impediment to taking that, you do a genomic profile into this much paperwork. How do you take that and give a meaningful interpretation to someone? And that's a major impediment to translating this science, and that's what we've spent months developing how this test is presented and reported. And then finally, we give a disease signature report. And that disease signature report is very extensive, very specific with the classes, types, and grades of various diseases that are included in there, acute and chronic durations, et cetera. It's not just cancer. It's very specific to the cancer, the type, the stage, ductal, lobal, whatever the case may be. And then the last page is a little bit more for the physician. And on those pages, which are hundreds of pages long, we've outlined the level of every single metabolite, and then known concentration ratios, so known levels of basically a healthful level, where in the literature it shows any person who expresses this metabolite below this level has certain health implications, and those who express this metabolite in higher than normal levels have this implication. Yeah, it's like a reference standard. So you have the first column will be the customers or your patients' metabolite levels per metabolite. The second column is what is considered too low. The third column is what is considered too high. And then we also give information on what that metabolite is, what pathway it's in. We give a citation for every single statement we've made. It shows, what the implications of low levels of that metabolite are going to mean, what the implications of high levels of metabolite would mean. So we've given a general description and implications of low and high levels for all 126 metabolites. And at the end, it's a very long report, but it's complete with, depending on the patient's personalized results, anywhere from 200 to 500 citations. And that's another thing that I think sets us apart from a lot of the entities in these spaces, which are run by an MBA, which is great, but we are scientists, and we want to make sure that scholarly rigor, scientific integrity, and ultimately transparency in how we report those results are always prioritized in every single Aristotle report we send out.
It's incredible the depth of insight that we get here. So there's a couple good questions from Martin that I wanted to bring up. One was the one you started to hit on, which is, how does this differ from the LCMS OAT? I'm probably pronouncing that wrong. And then he's looking for raw data on this.
Yeah, absolutely. So what we do is for when you say OAT, I assume you mean organic acids test. So for an OAT, we've completely obviated the need for an OAT, Martin. Every single panel, every single metabolite that you get in your OAT is included in the Aristotle. And it's actually the second most represented chemical class in our panel. And yes, I'd be happy to share with you raw data. We don't include raw data in the file. What we do is we include your concentration ratio. So the level that you are at compared to basically a database, an internal database that we have that is informed by 10,000 people who participate in the Human Metabolome Project, in addition to, when missing and needed, a genomic reconstruction of probable metabolite levels. And what we do is we measure your level to that average level that is deemed in the population, and we give you a concentration ratio, and that's your level of your metabolite. But we'd be happy to share raw data. And one thing that is very interesting for us for the blockchain idea is that raw data will be available to you immediately. There won't have to be a separate file we send to you. You can get your report that's generated, of course, with our AI, but the raw mass to charge file can sit on the blockchain. Your raw sequencing data from the metagenomic reconstruction will sit on the blockchain and will be accessible and completely transparent to you.
Fantastic. Do you see some other questions in here that you would like to answer, Paniz?
There's one question. I think we discussed how we differ from Viome in the sense that, do you want to continue a little bit?
Dietary supplements, I think, was a question.
Right. Also, another thing that we differ from Viome is that we're not selling you any. Yes, of course, you're given an action plan, Mallory. I see your question there. You're given an action plan in addition to the scores along those 12 health domains that we mentioned, the aging index, the mutational load. For each domain, relative to your score, you get specific instructions, and some might be, hey, keep doing what you're doing, and some might be, incorporate CoQ10 supplementation in your diet, incorporate XYZ foods, try eating at this time, changing your eating pattern, or maybe some nascent intermittent fasting protocols that we've also developed.
But what we're not doing is channeling you to a particular dietary supplement, which a lot of companies in this space are doing, in the spirit of what we're trying to accomplish here, and just being, scientific, I would say, we have no interest in pursuing that.
Yeah, we're not going to sell you supplements. That's another thing that I know sets us apart from other companies. We're not a supplement company. Our bottom line is not made off that. Our goal is to enhance scientific knowledge, medical practice, and ultimately put the power of that data into your hands. It is your data. We generally don't like even the term biohacking, because it always seems like you're trying to get somewhere you shouldn't be, somewhere you're not allowed, somewhere that is criminal for you to be at, like you're hacking something. But it's your own data. It's your own biology. And that is, we believe at Theriome, an inherent right for you to monitor and assess as you see fit. And our whole goal is to just give you those tools to do so with next generation platforms and next generation technologies. One thing, so how do we differ from Viome? Right, we're not an MBA, we are scientists. We don't sell you supplements. Our work is based on more than 120 papers that we've collectively published in this space. Not one published paper, I'm not going to say which entity, but one published paper and 3 white papers that have never been peer reviewed and only put on a preprint server. That's not a basis for a scientific company. And so we are not selling you supplements. We are scientists, and we are ultimately committed to the highest. We do not engage in, Michelle, we do not engage in selling data to third parties.
It's all protected.
Yeah.
It is. That's a major concern.
Yeah. I know Caitlin was saying something the other night, my wife, about doing a 23andMe test, and I was like, I don't want to sell it to the government, essentially. And she's, jokingly said, what are you, a serial killer? And I was like, no, I just don't want the government to have my stuff, So yeah, I can totally relate to that side of things. There's a couple other good questions in here too, though. One was, what about difficult to diagnose illnesses? Like, what value could somebody expect from this test in that space? And then kind of following up with that, Mallory asked, she's a friend too. So hi, Mallory, but are we given an action plan after we receive the results? So those 2 questions: difficult to diagnose, how does this help? And what does the action plan look like?
Yeah. So, so with the action plan, I'll just address that because we did, I think I just addressed Mallory's comment earlier, where I said, hey, we do provide, very detailed point-by-point recommendations based on the scores of your aging index. So some things could be if you have a 9. Yep, there we go. Yeah. So Mallory's got our answer. We do provide, just to reiterate, those personalized recommendations based on every patient's demographic and metabolomic data. So difficult to diagnose disorders. Extremely rare diseases, ultra rare diseases have actually become a rather fascinating thing for me lately. I'm writing a paper now where we analyze dental pulp stem cells from patients with FOP, which is a disorder known as fibrodysplasia ossificans progressiva. It affects currently 8 to 13 people, we think, worldwide, the whole world over. About a dozen people, no more, have this. And it essentially takes the connective tissue and it ossificates them to bone. And so these patients essentially die very early deaths, entombed in a prison of their own skeleton as bone restructuring and formation. Essentially, formation never stops and restructuring never begins. And it's a very rare disease. And we do not have currently the signatures of incredibly rare diseases in that panel of 344 confirmed blood signatures that we've curated for our test. It is, unfortunately, the treatment, the biomarkers that will come for those diseases will come later on as we enhance our testing, enhance our database, and we're able to mine that anonymized database for medical answers. We will continue to add disease signatures. Currently, in that list of disease signatures, the 344 signatures we query your metabolic profile against, those are mostly just related to the greatest contributors of human health. So early markers of myocardial injury, cardiovascular disease, late stage heart failure. We have Alzheimer's in there. Various cancers are in there at different stages and etiologies and grades. We have included in that panel behavioral outcomes such as autism, schizophrenia, depression. And those are the most typified ones, the most well curated, and the ones with the greatest evidence. But medical science, of course, is always adapting and evolving. So as our knowledge grows, we do aim to become more focused on diseases that are relevant to smaller populations, such as I'm talking about chronic illnesses, biotoxins. Sure, yeah, of course, yeah, more rare, yes, not the ultra rare diseases. Absolutely, our environmental exposure score is a great indicator of your exposure to molds and fungi. Our gut health is another great indicator of potential exposure. We also have various level metabolites. As you go through the glossary of your metabolites and you see these are associated with certain fungi and toxins. Many of them are included, and we've curated that panel with a specific eye to that. About fertility-related problems. So we can speak to some fertility-related problems molecularly for men, such as decreased spermatogenesis. For women, we do not have a disease signature in our panel currently. However, we do have a general reproductive health score. So that reproductive health score we can actually use to, for instance, Micah's assistant took the test, Maggie, and her reproductive health score was very low, meaning that she was not receptive to having another child. Turns out she was pregnant at the time she took the test. One of our investors gave me a test blinded, and he said, "I want you to tell if this is, my mom or my wife." And I was able to tell that it was his mom because the reproductive score was essentially not applicable. It was a sub-1 score. So I could tell this was a postmenopausal woman. And so there we have measures such as the reproductive score, which is for every person that takes the test, as well as specific metabolites related to specific processes in men only, such as spermatogenesis, or also gonad health and prostate health.
Yeah, and early detection of liver issues.
Absolutely. Liver health is a paramount thing. So, in the Persian language, the liver is so important that we don't say, "I love you with all my heart." We say, "I love you with all my liver." because that liver is essentially the producer of most of your body's enzymes. It is the central processing site for all of these metabolites mostly. And liver disease, liver failure, non-alcoholic fatty liver disease, cirrhosis. We also have liver cancers as part of our liver panels that are included, not just as the overall liver health, which is one of the 12 key health domains, but also various disorders that are included as part of those 344 blood disease signatures we query.
I do want to put in a little bit here. So Dr. Jasbi is trying to get the word out about this. He's from academia. He's not involved with a bunch of podcasts. He's not, going to dinner parties with Jeff Bezos yet. But we're trying to get the word out about this thing because it will have massive ramifications. And I'm actually helping out with this project. I'm sitting as the president currently to get this thing off the ground. And it's something I'm immensely passionate about. As you guys know, if you know my story, my brother Matt died of brain cancer. And I've had other family members die of cancer as well. But, the thing that goes on in my head is like, what if we were able to detect that at the earliest stage? What kind of difference would have been made? How much longer would he have lived? Would he have been able to go into remission? And I don't have the answer to that. But I do know that this test has so much potential that people will be able to detect things much earlier and improve their outcomes significantly. So what does that mean? That means that kids that wouldn't have had their parents or a mom or a dad will now have their mom or dad that would have otherwise passed from a certain chronic disease, or grandparents or brothers or aunts, uncles, that kind of thing. And so this really lights me on fire because it's an opportunity not just to deliver a test that's personalized and cool and cutting edge, but it's actually going to have massive ramification in terms of people's lives and being able to overcome these things that we're stuck with. And so it really falls in line with my mission in this world, which is to reduce suffering. If you guys have listened to me, I've talked about that a bit, but very, very excited about this. And so where this comes into play with you guys and getting the word out about this out there, if you have connections to podcasts, to people that can put Dr. Jasbi onto a platform to help promote this, please write us. That would be massively helpful. info@therio.me. It's right on the screen there. And just give us a connection, give us some information. And you don't owe us anything, but it's massively appreciated because that really helps in this mission of helping people with their health and transforming more from sick care into wellness. So any sort of connections that you guys have that can help us out would be massively helpful. Again, we're grassroots here. So we need your support, and you guys have been massively supportive of me and my business. And I am eternally indebted to everybody because of all the support that's happened. And I'm hoping we can do something similar with Dr. Jasbi here because he has something that is truly revolutionary. And things fail all the time. It's not easy. Like, this is not an easy project. So any sort of connection you have to a podcast or a platform or a radio show or a TV show or morning news, whatever it is, Dr. Jasbi told me that he is happy to jump onto it. So please write us at info@therio.me. If you have any sort of connection like that and just connect us with the person, we'd be happy to set up a meeting before jumping onto a podcast or something like that. And in addition, we're going to be closing this out now because we got to get running. We got science to do, right? So it's exciting. But
if I didn't answer any questions, please write us info@therio.me, as Micah said, or if you want to ask any questions of me directly, I should have put my own email up here, but it's Jasbi, my last name, jasbi@therio.me. I'll put it here in the chat. And
You already got it.
there we go. Yeah. So please feel free to email me and I'll field questions as they come in. Thank you guys all for your interest in attending this webinar on behalf of,
I'm going to give one last chance here, and then we'll spend another 60 seconds or so. If you're interested in this test and want to get a quote and instructions on how to do it, again, it's $331 off. It's normally $830. It's down at $499. And there's 40 of these tests available. He has the material for 40 of the tests before he has to reorder, and that'll take some time. So if you want to get on that train, just drop your email into the chat. A quote with instructions will be sent to you. Makes it super, super easy if you're interested in doing this test. So that's the way to do it. And any closing remarks that you have, Dr. Jasbi or Alex? Alex, the reason I'm not saying doctor, is it Moore? Is that how you pronounce your last name?
All right, it's pretty simple. Yeah.
Moore is
Okay,
spelled
Dr. Moore.
M-O-H-R.
I'm sorry. I keep on going between Dr. Alex and Alex because I'm going to mess up his name. So Dr. Moore, any closing remarks?
Thank you, guys. Thank you for attending the webinar, and thank you for sharing the vision. Thank you for your insightful questions. And if there's anything I didn't address, please email me. I'm happy to discuss more with you. Alex, anything?
Oh, it was great. We have a lot more in the future planned, so we're very excited.
Hope you guys keep your eye on this space. Yep.
Yeah,
We'll talk
keep
soon.
your eye out for sure, because this is going to be a big thing within a couple of years. And thank you so much, everybody. Massively appreciated that you took time out of a Wednesday afternoon to discuss with us and have a conversation. I hope you guys have a splendid day and fantastic conversation. Thank you, everybody.